muscles and fascia. See here, these are the vertebral column, lumbar vertebral column mainly, T12 to L5, and their transverse processes, here 3, all these muscles, up to the pelvic ring, it is forming the posterior abdominal wall. Okay. So it is strong and stable because it is constructed by bones, muscles and fascia, and it supports the retroperitoneal organs, vessels and nerves. Posterior abdominal wall consists of mainly 3 parts: bony part, muscular part and fascia. Fascias are, these 3 types of fascias are present: psoas fascia, fascia iliaca and thoracolumbar fascia. We will discuss this bony and muscular part in details. Next, so muscles of the posterior abdominal wall, 3 muscles are present. Psoas major, see here, this one is the psoas major. Iliacus, here in the iliac fossa of the hip bone, this is iliacus. Iliacus and psoas major together forming the iliopsoas tendon, and it is inserted over the lesser trochanter of the femur. Then quadratus lumborum, here is the quadratus lumborum, here is origin from the 12th rib, and here is the insertion of the quadratus lumborum. And sometimes psoas minor is present in some cases. Okay, very rarely present psoas minor. Next, see these muscles. See, this is pelvic brim and this is 12th rib. Okay, so here is the quadratus lumborum. This is psoas major, here is iliacus, and this is psoas minor. These are the 4 main muscles of this posterior abdominal wall. Next, so we will read the individual muscles, their origin, insertion, nerve supply and action. So first, the psoas major muscle. Origin is of the muscle arises from 14 fleshy slits, and when the fleshy slits are arising, 5 slits from the intervertebral discs between the T12 to L5 vertebra and adjoining margins of the bodies of this vertebra. 5 slits from the anterior surfaces and lower borders of the transverse process of 5 lumbar vertebra, and 4 slits from the tendinous arches that bridges the constricted sides of the bodies of lumbar vertebra. So total 14 slits of origin is present for psoas major. Insertion, lesser trochanter of the femur via the iliopsoas tendon. What are the functions? Acting from above, it is chief flexor of the thigh at the hip joint. And acting from below, it flexes the trunk on the thigh, as in raising the trunk from recumbent to sitting position. So these are the functions of the psoas major. Now what is innervation? Ventral rami of L1 to L3 nerve. Okay, so this is innervation. Next, so here is the psoas major, you can see origins from this transverse process and the tendinous arches connecting the vertebral bodies and this intervertebral disc. From all these are the origins, and here is the insertion in the lesser trochanter via iliopsoas tendon. Next we go to psoas minor. It is present in about 50% individuals. Okay, not in all the individuals it is present. Origin, it arises from the side of intervertebral disc between the T12 and L1 here. From the intervertebral discs between T12 and L1 and adjoining parts of their bodies. Insertion into the iliopubic eminence. Okay, here iliopubic eminence is here. Here is the insertion of psoas minor. Next is the nerve supply. It is supplied by branch of L1 spinal nerve. Action, it is a weak flexor of the trunk, small muscle. Okay, next is iliacus. See, in the iliac fossa, there is iliacus muscle. It is a fan-shaped muscle and forms the lateral component of the iliopsoas muscle. Origin, it arises from the upper two-thirds of the floor of the iliac fossa, inner lip of the iliac crest, and upper surface of the lateral part of the sacrum. This is the origin of iliacus. Insertion into anterior surface of lesser trochanter and in area 2.5 cm long below it here, extend towards the shaft also. In the lesser trochanter, extending towards the shaft on the posterior surface. Action is, nerve supply is femoral nerve. Iliacus is supplied by femoral nerve. Action, it is helps in flexion of the thigh and the lumbar part of the vertebral column. So this is action of the iliacus. Next, so three muscles we have discussed. This is last muscle, quadratus lumborum. All these muscles you will see in your posterior abdominal wall dissection. Okay. So quadratus lumborum origin is from posterior one-third of the inner lip of the iliac crest. Here is the origin. Posterior one-third of inner lip of the iliac crest. And lower 3 to 4 transverse processes of lumbar vertebra here. 3 to 4 lumbar vertebra. Insertion is medial part of the anterior surface of 12-3. Here is the insertion. Medial part of inner anterior surface of 12-3. And upper lumbar transverse process. Here is one insertion. Now, where is the nerve supply? Supplied by ventral rami of T12 to L3 or L4 lumbar spinal nerves. What is the action? It is a lateral flexion of the lumbar vertebral column and extends the lumbar vertebral column. So this is action of quadratus lumborum. Here is the position. You can see it can act as a lateral flexor. Next. Now, on the posterior abdominal wall lies the great vessels, the abdominal aorta and the inferior vena cava. So, the great vessels of the abdomen are these two, abdominal aorta and inferior vena cava. Next, so abdominal aorta, it is continuation of the descending thoracic aorta at the aortic orifice. T12 level lies the aortic orifice. So it is continuation of descending thoracic aorta at the aortic orifice of the diaphragm opposite to the lower border of T12 vertebra or intervertebral disc between the T12 and L1, and it descends vertically downward and slightly to the left in front of the vertebral column and terminates in front of the lower part of the body of L4 vertebra and dividing into right and left common iliac arteries. Okay, so this is the course and termination of abdominal aorta. Next, so these are the branches we have discussed in previous class. So anterior branches are three branches: celiac trunk, superior mesenteric artery, inferior mesenteric artery. Celiac trunk, superior mesenteric, inferior mesenteric. Then lateral branches, these are paired branches, and these include this inferior phrenic artery. Here is the green one are the lateral branches. Inferior phrenic artery, middle suprarenal artery, renal artery, and gonadal, testicular or ovarian. Testicular for male and ovarian for female. Okay, so these four are the lateral branches. Next posterior branches. There are four pairs of lumbar arteries. This small one, these are the four pairs of lumbar arteries, and here is median sacral artery. So these are posterior branches. Next is terminal branches, two common iliac, right and left common iliac arteries. So these are the branches of the abdominal aorta. Okay, maximum branches you will see in your posterior abdominal one dissection class. Okay, next. Okay, so three unpaired ventral branches to the gut, three paired lateral branches to three paired glands, suprarenal gland, kidney and gonads, paired posterolateral branches, and above the aorta also gives two paired inferior phrenic artery and three terminal branches. Okay, median sacral is also considered here terminal. Okay, next we go to the veins. Inferior vena cava. Inferior vena cava is the largest and widest vein of the artery, and it drains most of the blood from the body below the diaphragm into the right atrium of the heart. What is its formation, course and termination? The IVC is formed by the union of right and left common iliac veins. Here, right and left common iliac veins join to form the IVC. কি কথা বলছিস এত কন্টিনিউয়াসলি কথা বলেই যাচ্ছে কিছু জন So IVC is formed by union of two common iliac veins in front of L5 vertebra. Vertebral level is important, I have told. Below the aortic bifurcation. Aortic bifurcation occurs at L4, okay, and IVC formation occurs at L5. And behind the right common iliac artery it occurs. Because IVC lies on the right side and aorta on the left side. Okay. So, and what is the course? It ascends in front of the vertebral column on the right side of the aorta. It then arches forward on the right crus of the diaphragm to reach the groove on the posterior surface of the liver. And it pierces the diaphragm at the level of T8 vertebra, terminates by the right atrium of the heart. Okay. Next, these are the tributaries. IVC receives these tributaries, three formative tributaries, two common iliac vein, one median sacral vein, three abdominal wall tributaries, inferior phrenic vein, third and fourth lumbar veins. The first and second lumbar veins end in the ascending lumbar vein. Okay, and three lateral visceral tributaries, right and left renal veins and suprarenal veins, right and left testicular or ovarian veins, and three anterior visceral veins are the right, middle and left hepatic veins. Okay. So these are the tributaries of the IVC. IVC and abdominal aorta very frequently given in your OSP, and you have to know the branches of aorta and tributaries of the IVC. Very frequently it is asked in your Viva, also in OSP. Okay, very important question. Next, there are presence of lymph nodes. These are located along the external iliac arteries, common iliac arteries and abdominal aorta. Here, see the green one are the lymph nodes. Another important lymphatic tract is present, that is cisterna chyli. Okay, and the lymph nodes lying in front of the aorta is pre-aortic, and on the lateral to the aorta is lateral para-aortic lymph nodes. Okay, and these are the common iliac, external, internal iliac group of lymph nodes are present. Accordingly, they are termed as external iliac, common iliac and aortic lymph nodes, according to the vessel along which the lymph nodes are lying. Next is nerves of the posterior abdominal wall. There are subcostal nerve. It is the ventral ramus of the thoracic spinal nerve. Here, this is subcostal nerve. Okay, thoracic spinal nerve, ventral ramus. And there is lumbar plexus and its branches. See, these nerves are joining to form this lumbar plexus. Lumbar plexus is formed by the union of ventral rami of lumbar plexus is very frequently asked in your Viva. Its branches and its root value of all these branches. Lumbar plexus is formed by the union of ventral rami of L1 to L3 lumbar nerves and a larger upper part of the ventral rami of L4 here. L1, L2, L3 and larger part of L4, and smaller part of L4 is going to join with the sacral L5 and other sacral nerve forming the sacral plexus. Okay, next. अच्छा आगे previous आगे okay, the lower smaller part of the ventral ramus of L4 nerve joined with the ventral ramus of L5 nerve to form the lumbosacral trunk. See, smaller part and L5 joined to smaller part of L4 and L5 joined to form the lumbosacral trunk, and which takes part in formation of sacral plexus. Okay. Next, now we will discuss the branches of the lumbar plexus that also you will see in the lying over the posterior abdominal wall. Okay. So first is iliohypogastric nerve. Yesterday you have seen in your inguinal region iliohypogastric. Okay, so iliohypogastric nerve emerges at the lateral border of the psoas major, and this lumbar plexus it lies within the content of psoas major. Within the muscle psoas, within the psoas major muscle, the plexus is formed, and the nerves are coming out from the lateral and medial border of the psoas major muscle. Okay, so maximum branches are coming out along the lateral border. So iliohypogastric emerges at the its root value is L1. Root values are important. Emerges at the lateral border of the psoas, runs downward and laterally in front of the quadratus lumborum and behind the kidney and colon, pierces the transversus abdominis a little above the iliac crest and runs in the abdominal wall muscles. Okay. Next is ilioinguinal nerve. It has the same force as iliohypogastric nerve but on a slightly lower level, parallel to the iliohypogastric, below the iliohypogastric. Okay. L1, root value is L1. Then is genitofemoral nerve. Its root value is L1 and L2 ventral division. Emerges on the anterior surface of the psoas major near its medial border and runs downward in front of the muscle. It is generally plastered with the psoas major on its anterior surface. That's how you identify this nerve, genitofemoral nerve. Okay, it is also given in your OSPE, genitofemoral. Near the deep inguinal ring, it lies in front of the external iliac artery and divides into two branches, femoral and genitofemoral. Okay. Genital branch of genitofemoral, it is a part, it lies within the spermatic cord, and femoral branch enters through the lateral compartment of femoral sheath. You have learned both of this. Next is lateral cutaneous nerve of the thigh. Okay. Two branch, medial and intermediate cutaneous thigh, intermediate cutaneous nerve of thigh. Those two were branched from anterior division of femoral nerve. But lateral cutaneous nerve of thigh, it is a direct branch from the lumbar plexus. So its root value is L2, L3 dorsal division. Emerges at the lateral border of the psoas, then runs downward and laterally across the right iliac fossa over the iliacus and reaches the anterior superior iliac spine. It lies near the anterior superior iliac spine. Then femoral nerve root value is L2, L3 and L4. It is formed by the dorsal division of these nerve roots. Emerges at the lateral border of psoas below the iliac crest, runs downward and slightly laterally in the furrow between the psoas and iliacus, and it lies under cover of the fossa iliaca. That means iliopsoas groove. Femoral nerve lies in the iliopsoas groove and it lies under cover of the fossa iliaca. Next is obturator nerve. L2, L3, L4 ventral division. Femoral nerve was dorsal division, এখানে ventral division, obturator nerve. Okay, these divisions are also important. Ventral rami or the dorsal rami. So it emerges on the medial side of the psoas major. It is the only nerve emerging from the medial side. That's why how you can identify. Very frequently asked in your viva, where will you find the obturator nerve? Okay. So it emerges on the medial side of the psoas major muscle and runs forward and downward on the pelvic wall. It is forming oil structure. During sacrum you have read oil structure. From outside inward over the ala of the sacrum lies the wall structure. O is formed by obturator nerve, okay, iliolumbar artery, lumbosacral trunk and sympathetic trunk. All these four were forming the wall structure. So it is emerging on the medial side of psoas major, you have to remember. And then the lumbosacral trunk, that is L4, L5 ventral rami. It is formed by union of the descending branch of nerve L4 with nerve L5. We have read smaller branch from L4 and L5 roots joined to form the lumbosacral trunk. It is related medially to the sympathetic chain and laterally to the iliolumbar artery and the obturator nerve. That wall structure. Okay. O is lateral, obturator nerve lateral, then iliolumbar artery, then lumbosacral trunk, then sympathetic chain from lateral to medial. Next. See here the nerves. Okay. So this one is the lateral femoral cutaneous nerve going towards the anterior superior iliac spine, and this is psoas major. Plastered over the psoas major will be the genitofemoral nerve. Okay. And below the iliac crest from the lateral border, thick nerve is the femoral nerve, and along the medial border comes out the obturator nerve. And here is the sympathetic chain. So all these are branches from the lumbar plexus. Okay, and here is the formation of hypogastric plexus from the greater and lesser splanchnic nerves. Okay, so all these nerves you have to see during posterior abdominal wall dissection. Next. These are the nerves near the 12th rib, subcostal nerve, root value is T12. Then from the L1, iliohypogastric above, ilioinguinal below. Then genitofemoral nerve, lateral femoral cutaneous nerve, femoral nerve, here is the obturator nerve and lumbosacral trunk. All these are main important branches of lumbar plexus. Next. These are the formation of plexus. Okay. Superior mesenteric, celiac plex, ganglion, superior mesenteric, inferior mesenteric plexus. These are formed by the sympathetic chain branch. Okay. Next. Now clinical aspects. Okay. Psoas abscess. Psoas first, iliacus and psoas major. These two are covered by a single fossa, that is known as psoas fossa. Okay, and the fossa covering iliacus is sometimes called fossa iliaca. So psoas fossa forms a long tubular sheath, osseofibrous tunnel called the psoas sheath, extending from the upper lumbar region of the posterior abdominal wall to the groin here. This is psoas major muscle from the upper lumbar region, T12 থেকে start হচ্ছে L1 to here up to the groin region, extending from upper lumbar region to the posterior abdominal wall to the groin. And tubercular infection of vertebra of the thoracolumbar region causes destruction of their bodies, leading to formation of an abscess, and the pus cannot spread anteriorly due to anterior longitudinal ligament. Therefore, it spreads laterally into the psoas sheath, forming the psoas abscess. Okay. And the pus can also enter the psoas sheath from the psoas posterior mediastinum through a gap deep to the medial arcuate ligament, and pus may then spread downward along the psoas muscle under the inguinal ligament into the femoral triangle, where it protrudes as a soft swelling. Okay, so our psoas, under the psoas sheath, pus can come from the vertebral bodies if there is tuberculous infection, and also from the posterior mediastinum. During thorax you have read, from the posterior mediastinum pus can trickle through the psoas sheath and pass into the femoral sheath. So it can extend up to the thigh region. Pus can extend up to the thigh region and form a soft swelling over the thigh. Next. Next is aortic aneurysm. Okay, aorta is main important vessel of the posterior abdominal wall. So aortic aneurysm is localized dilatation of the aorta, commonly occurs below the origin of renal artery. Here the renal artery there is dilatation, that is aortic aneurysm. In 95% cases below the renal artery. Usually occurs in elderly men. Okay. Most common cause of aortic aneurysm is atherosclerosis, which weakens the aortic wall, and clinically it presents at pulsatile and expansile abdominal mass, typically just superior to and to the length of the umbilicus. Okay. Next there is compression of inferior vena cava. Inferior vena cava is commonly compressed by an enlarged uterus during the last trimester of pregnancy, and this causes edema of the ankle and feet because venous drainage is not occurring properly. So there will be edema of the feet and ankle and varicose veins in the lower limb. Okay, most commonly occurs in pregnancy, varicose vein, and the compression and blockage of the IVC by the malignant retroperitoneal tumor result in dilatation of the anastomotic channel between IVC and SVC, superior and inferior vena cava. Anastomotic channels, caval shunt, so that the blood would be returned to the right atrium. Okay, and clinically it presents as the prominent subcutaneous veins called thoracoabdominal vein formed due to dilatation of anastomotic venous channel between the lateral thoracic vein, a tributary of axillary vein, tributary of superior vena cava, and the superficial epigastric vein, which is a tributary of femoral vein, and femoral vein is the tributary of IVC. So in between tributary of SVC and tributary of IVC, there is formation of anastomotic channel. SVC tributary ki holo this lateral thoracic vein, okay, which is tributary of axillary vein. Axillary vein is tributary of SVC, okay, and here from below it is from the superficial epigastric vein and tributary of femoral vein. Superficial epigastric vein you have seen during the inguinal region. In the superficial fossa it is lying, okay. Superficial epigastric vein lies in the subcutaneous plane, lying along the branch of femoral, superficial branch of femoral artery, superficial epigastric vein. Next, okay, that's all. Any queries regarding this topic? Query answer issue? Okay, so you have to read the muscles. Okay, individual muscles can be given in your OSPI question. IVC, SVC, IVC, abdominal aorta. These two are very frequently given, and the lumbar plexus and its branches. Okay, you have to know the root value. Individual nerves can be given in your OSPI. Okay, a lumbar plexus can come as a short note. Abdominal aorta, IVC can come as a short note, okay, and posterior abdominal muscles like this, it can come as a short note, okay.
It has been a practice in many medical colleges to keep weekly one exam each subject, so that whatever you study in the week, you get a time to recapitulate. Mostly people don't pass, they fail, doesn't matter. But it's not the result of the race, but the participation in the race which matters. Because at the end of the year, the small preparation accumulate to help you a lot. Correct? So with that in my view, I want to keep an examination with your permission. Suppose I keep an examination on Wednesday, 6th, the next day is our examination, my supplementary practical examination. So we will be busy for 2 more days, 2 to 4 days, and you do not turn up. Only 10 or 12 students come to examination. then it will be very heartbreaking for us. Correct? So that's why it's my opinion. If you agree, then only we'll keep the examination, or else we'll keep normal class. So I would request you to open this one first. All of you, open this diagram, because I am going to change it. You have to see it from your own copy. Fine? Ready? So now we will see the sequential stepwise biosynthesis of new fatty acid. The new fatty acid is synthesized from the acetyl coenzyme A, produced or supplied by not beta oxidation but oxidation of glucose. Remember, acetyl coenzyme A, which is fed into TCA cycle to run the show means to produce energy, can be produced by two ways, either one glucose oxidation or fatty acid oxidation. Now these two will never take place simultaneously. When glucose is plenty abundant, then fatty acid oxidation will stop. When glucose is absent, fatty acid oxidation will start. These two will never occur together simultaneously. So when I say fatty acid synthesis, that means beta oxidation is stopped. Only glucose is abundant, plenty of glucose is there. I am eating couple of plates of biryani. 2 plates, 3 plates of biryani, or a lot of rice, a lot of carbohydrate, a lot of French fries, whatever it is, and that is generating huge amount of carbohydrate. This excess carbohydrate is stored in the form of fat, triglyceride, correct? The triglyceride. For that, we have to synthesize fatty acid, and glycerol will be supplied by. Triglyceride is made up of two things, fatty acid and glycerol. So fatty acid will be supplied by acetyl coenzyme A. Glycerol will be supplied by glycolysis. Both are coming from carbohydrate. Yeah? Now we'll go to sequential part. Now here, if you can recall, in beta oxidation, the reactions were oxidation, hydration, oxidation, cleavage. Now just reverse the paper. What do you get? Reverse the paper. Oxidation, oxidation, hydration, oxidation, cleavage. Now just reverse. What is there? Cleavage. Cleavage. What is the reverse of cleavage? Joining, condensation. Correct? Then reduction, then dehydration, then very good. You can go through like this, or you can read the slide also. Correct? So this we have to remember. Wherever there was dehydrogenase, this time it will be reductase. Correct? Wherever water was added, structure, see the structure. This is the first step, formation of malonyl CoA, number 1 step. Like beta oxidation, it was first what? Activation of fatty acid, then transport across the membrane, then beta oxidation proper. Similarly, in this case also, this is taking place in where? Cytosol. So, first step is formation of malonyl CoA, then de novo synthesis proper. First step is formation of malonyl CoA, which is formed by carboxylation of acetyl coenzyme A to form malonyl CoA. What is the structure of malonyl CoA? It has 2 carboxylic group. Remember, the organic acids containing 2 carboxylic groups, 2 carbon compound, oxalic acid, 3 carbon compound, malonic acid, 4 carbon compound, succinate, 5 carbon compound, butyrate, correct? 6 carbon compound, adipate. You get it? Ohm's gap, +2 formula to remember the dicarboxylic acids. OMSGAP, right? So now this malonyl CoA is formed with the help of ATP. Every step it will require. As a matter of fact, to synthesize a palmitic acid, if you can recall, I told that every time a palmitic acid is formed and then it is elongated to form other fatty acids. So base fatty acid molecule should be palmitic acid. So what is a palmitic acid? 16 carbon compound. So how many acetyl coenzyme is required? 8. Now out of 8, 1 will be required as acetyl coenzyme A and other 7 will be converted to malonyl. This is the most important point in this particular step. You remember, it is not very easy. Out of 8 acetyl coenzyme A, 1 will be required as acetyl coenzyme A and rest 7 will be required as malonyl coenzyme A. Why? Because it has to transfer and it requires energy to transfer a molecule from one place to another. Correct? Now, acetyl coenzyme A, if transferred in the form of acetyl coenzyme A, then it will lose energy and it will be less than something called acetyl coenzyme A. But if we increase the molecule to malonyl coenzyme A and then we transfer, the reduction of energy or the dissipation of energy will remove some part of malonyl coenzyme A and acetyl coenzyme A will be added. তাই না? যদি আমি 100 টাকা নিয়ে জার্নি শুরু করি তাহলে জার্নির শেষে আমার কাছে 100 টাকাই থাকবে, কিন্তু আমাকে যদি 100 টাকা ট্রান্সফার করতে হয়, যেমন আমরা কি করি, কি বলে মানি অর্ডার করি যখন, if we want to transfer 100 rupees, do we pay 100 rupees or we pay more? We pay more because এই 100 টাকা একটু বেশি না দিলে 100 টাকাটা পৌঁছাবে না, তাই না? তাই আমি অ্যাসিটাইল কোএনজাইমটাকে প্রথমে ম্যানুয়ালি করে আনলাম, সেটাকে ট্রান্সফার করলাম। Clear? To increase the energy initially for the transfer purposes. Now, why do we transfer? Now, these steps. Now, it is very, very difficult. Slowly, you try to understand. Just try to understand. I hope this will never be asked in your final examination. Hope. Nowadays, they don't ask, they try to keep it away. But at all, if it comes, then it will be difficult. Only the thing is, understanding is important, stepwise. Now, keep your diagram open of the fatty acid synthesis complex and concentrate on the name of the enzymes. The first step in the enzyme is existed in that complex form where subunit division and functional division is predominant. So, one side will be producing one palmitic acid, another side will produce another palmitic acid. Now, so consider one side here, other side is just like a mirror image. So, consider the reactions taking place in the one side of the functional division. You find the first step, acetyl-CoA transacylase or acetyl-CoA transferase. Now, this enzyme will transfer one molecule of acetyl group to carrier protein, acyl carrier protein. Whatever new thing will be added to that enzyme complex will be added to ACP. Whenever you enter to a restricted place, you face police. So, at least one ACP level police will be there, police officer, to greet you whenever you try to enter into any restricted area. Just remember like this. So, the Assistant Commissioner of Police will receive the first molecule of acetyl coenzyme A and then it will transfer the acetyl coenzyme A to the cysteine SH. Which cysteine SH? The cysteine SH of the other polypeptide chain. First one is transferred to one pan SH, then it is transferred to the cysteine SH of the other polypeptide chain. In your diagram, the left side, you have one cysteine SH above and one pan SH below. Am I right? Huh? So, it will transfer the acetyl coenzyme A to pan SH first, and then something which is unknown will transfer that acetyl coenzyme A from pan SH to cysteine SH above the line, subunit division. So, from one polypeptide to another polypeptide, this transfer will take place. Clear? Now, every time this will take place, every step. Okay? One subunit will be functioning, another subunit will be catalyzing. Done. In the next step, malonyl transacylase, which is the third enzyme. Now see, the first enzyme is not yet come to action. First enzyme ketoacyl synthase. We are starting with second, then third enzyme. See that diagram. Second enzyme, acetyl transacylase. Third enzyme, malonyl transacylase. So acetyl transacylase, we can say, transfers one acetyl group to 16SH to make it more easy to remember. Acetyl transacylase transfers one acetyl group to 16SH. Malonyl transacylase will transfer the malonyl group to ACP, pan-SH. Actually, it goes through pan-SH only, the acetyl coenzyme. But to remember more easily, we can omit that this small step we can omit. Here to here. Many of the books have done this to make it more easier for you to understand. Actually, the sequence of events is like this. But many of the books have given the diagram after this one, not this one. Omitted this step. Okay? So you can remember this also, we won't mind. So for your sake, let us consider acetyl transacylase is transferring one acetyl group to 16SH, and malonyl transacylase is transferring one malonyl group to pan-SH. Clear? This step, up to this step is there. Done? So now we have an enzyme. This is one subunit of enzyme, which is another subunit of enzyme. One subunit of enzyme contains acetyl group, another subunit contains malonyl group. So we call it acetyl malonyl enzyme. Acetyl malonyl enzyme. Enzyme is bound to both. In the next step, remember the color. This is somewhat violet and this is red. Huh? It a battery change for. Now tell me. Now this reaction 4, ketoacyl synthase. Which enzyme? First enzyme. First enzyme. So now enzyme number 2, then enzyme number 3, then enzyme number 1. Right? What is it doing? Transferring some molecule to one place to another place. Which one is transferring to whom? See and tell me. দেখে বল কোন গ্রুপটা কোথায় ট্রান্সফার করছে? অ্যাসিটাইল গ্রুপটা ম্যানোনাইল গ্রুপে আসছে না ম্যানোনাইল গ্রুপটা অ্যাসিটাইল গ্রুপে উপরে আসছে? Easy to remember, the growing fatty acyl chain requires a carrier. Which side is the carrier protein? ACP. So the acetyl group will come to malonyl group. Malonyl group will not go to acetyl group. Why? Because is newly added carbon dioxide. It was not a part of the acetyl coenzyme derived from citrate. The carbon dioxide of malonyl CoA, the third carbon, is newly added. That is not a part of glycolysis end product. So that is added to provide energy. At the destination, it is removed. So to do so, we have to transfer the acetyl group onto malonyl group. The smaller one will jump onto the top of the bigger one while removing one of the carboxylic group. So we added one acetyl carbon dioxide, we made it malonyl CoA, then we removed it after its work is done. So now we get 2 plus 2, 4 carbon compound attached to ACP. and both are having keto group. So what do we call? Beta keto acyl enzyme. Beta keto acyl enzyme. Now this is the joining part. Joining part. First reaction catalyzed by the first enzyme, ketoacyl synthase. Correct? What is the next step? Hydratase. Hydratase. Or dehydratase. So it will remove water. The next step, water is removed to form this one, beta hydroxy acyl enzyme. Beta hydroxy acyl enzyme. This was keto, beta keto acyl enzyme, then beta hydroxy acyl enzyme. Then beta hydroxy acyl enzyme will undergo reduction. That means withdrawal of hydrogen ion. NOL reductase to form this one. These two hydrogen will be removed and it will form this one. Followed? So now it forms a simple aliphatic chain attached to what? ACP. The fourth step is done. Joining, reduction, dehydration, joining. Now the next cycle will begin. The next cycle will begin only when there is a vacant seat for malonyl coenzyme A. Because we have only one acetyl coenzyme A and 7 malonyl coenzyme is waiting. Only one we have used. Rest 6 are waiting for their turn. So seats has to be vacant for malonyl CoA. Where does malonyl CoA sit? With ACP. So the whole structure will be transferred from ACP to 16SH. ACP to 16SH. ACP to 16SH. So how many times we are transferring? 3 times. 3 times we are transferring. First, the acetyl group, it first joins with ACP, then transfer to 16SH. Next, acetyl group comes to malonyl CoA on top of malonyl CoA. Second transfer, third transfer, it continues the synthesis, the reduction process, and then the whole aliphatic chain is transferred from pan-SH to 16SH. And then next cycle begins with a fresh malonyl CoA. Like these 7 cycles takes place and we get at the end palmitic acid. The moment palmitic acid is formed, all the subunits get disintegrated. All the subunits gets disintegrated. They go away from each other and palmitic acid comes out of the structure. Followed? Like this it goes on happening again and again. This process is very fast, rapidly they can synthesize. So we become very fat, we become fat very fast. If the process is very slow, then we could not grow our weight. The process is very, very fast, very rapid. Done. Any question? Quickly. So far, just remember the sequence of events and keep the fatty acid synthase complex in front of you. First, you remember the names of the fatty acid synthase complex enzymes. It made up of 7 enzymes and 1 carrier protein. Okay? And then 7 of the acetyl coenzyme is converted to malonyl CoA by acetyl CoA carboxylase, which require ATP and biotin like any carboxylase reaction. Fine. Then one malonyl CoA is transferred to pan-SH, one acetyl CoA is transferred to 16SH, then acetyl CoA is transferred to malonyl CoA to form beta keto, then one reduction is done. So 2 molecules of NADPH will donate the 2 molecules of hydrogen and 2 atoms of hydrogen. So it will become beta hydroxyacyl CoA, then it will become beta keto, then it will split into, again dehydration will take place, it will make this one, aliphatic chain, palmitic acids. But all joining with ACP. The last step, the whole molecule is transferred to 16SH, keeping the pan-SH free for next malonyl-CoA molecule. Clear? This is the order of sequence. You remember this, that's all. Fine? Now let us move. Now we have in our body many fatty acids which are longer than palmitic acid. How to synthesize them? We have one brilliant elongase system present in microsome. What is microsome? Anyone? What is microsome? We have microsomal chain elongation system. What is microsome? Microsome is the other name of endoplasmic reticulum. Endoplasmic reticulum in vitro is called microsome. Endoplasmic reticulum in vitro is called microsome. So enzymes present in endoplasmic reticulum should be present in microsome, in the test tube. When we break the cell, the endoplasmic reticulum, they coalesce to form sac-like structure, retaining all the contents of endoplasmic reticulum within the sac. This particular sac, which is formed artificially during the lysis of the cell, is termed as microsome. So if we want to examine all the enzymes present in the endoplasmic reticulum, we need to separate the fraction in the form of microsome first, and then we can run the experiment. So microsomal chain elongation system actually takes place within the endoplasmic reticulum. Now these chain elongation system, we have precisely two types of, but broadly, not precisely, broadly two types of enzyme. One is reductase, another is desaturase. Microsomal chain elongation system contains broadly two types of enzymes. One is reductase, another is desaturase. Desaturase is what? Which introduces double bond, desaturate, right? And reductase means which introduce hydrogen ion, H plus atom, right? H plus, or a hydrogen atom, not plus, hydrogen atom. What is the name of the reductase? NADH, cytochrome B5 reductase. Write it down. Microsomal chain elongation system is contains two sets of enzyme. One is NADH, cytochrome B5 reductase, cytochrome B5, beta choto hate, small b, cytochrome B5 reductase, and number two is desaturase. Now different animals have different types of desaturase. Different animals have different types of, like for example, we do not have desaturase below delta 5. That means we cannot introduce the double bond below 5th carbon. If we could do that, then we need not to rely on essential fatty acids. We could synthesize our own. Because we do not have any desaturase system beyond delta 5 desaturase, that's why we cannot synthesize essential fatty acids. Essential fatty acids, which are essentially essential, are of two types. What is? Alpha linoleic and linolenate. To remember the spelling, this way we pronounce it: linoleic, linolenic. Now there are different forms: alpha, beta, gamma. So the alpha form is essential, not gamma form is not essential. That we can synthesize. Alpha form is essential, right? So dihomo-gamma linolenate can be synthesized, but linolenate cannot be synthesized. So these two becomes highly essential. Clear? Why? Because we do not have desaturase enzyme beyond delta 5. Beyond delta 5. Just remember this line. This line is very, very important for 1 mark question. For 1 mark question, delta 5 desaturase. We do not have any enzyme below delta 5 desaturase. If there any other animal who are having it, for them linoleic acid is not essential. They can synthesize their own. Fine. This is the sequence of events taking place in the elongation for reduction, not required to remember. Do not remember. Do not remember. Okay. And this is the system how it occurs. See, it starts with palmitic acid. Only desaturase is acting. It forms 16:1 delta 9 palmitoleic acid. Then if you want to elongate it, then we send it to where? From mitochondria to, from cytosol to, hepatocyte cytosol to, it is in cytosol of hepatocytes. Now from cytosol to endoplasmic reticulum or microsome. Inside microsome, it will be elongated to stearic acid. How? Elongase enzyme will add one molecule of acetyl coenzyme A to the palmitic acid. So it will become 16 plus 2, 18. Then it will add on desaturase. It will introduce one double bond. Here one double bond, 9, 12. If it goes this side, it becomes linolenate. If it becomes this side, it becomes arachidonic. Correct? Similarly. Now these reactions will not take place unless we have delta 4 desaturase. We do not have delta 4 desaturase. We have up to delta 5. Delta 4, 3, 2 এগুলি আমাদের নেই. So we cannot synthesize linoleic acid or এগুলি করতে পারি না. Clear? Now we'll go to the regulation part of this part. The regulation, remember, this particular pathway is regulated by hormones as well as covariant modification. Now, for everything, this is a part of metabolism. So, it has a short-term and long-term control. Like slowly, when we come to the college, we were thin. By the time we finish the college, we become fat. So, throughout the 5 years, we gain more knowledge and more weight. Right? When you look back after passing MBBS from this college after 5 years, look back to your first year photograph, you won't be able to recognize yourself. So many things will change. Boys will lose hair, hairs will become grey because of us. We also lost because of someone else, and we are responsible for your hair loss for next 5 years. Correct? So this will happen. So this will happen. Similarly, the long term is what we can manage through change in the lifestyle. That you forget. Short-term control and long-term. Short-term control can be divided again further in allosteric modulation. একটা নেটওয়ার্ক করে নাও ছোট্ট করে. Short term, long term. Short term further divided into allosteric and covalent. Allosteric and covalent. Hello? Allosteric regulation. What we are having? The concentration of citrate, number 1. The concentration of citrate. কি করে citrate? Citrate allosterically stimulates acetyl-CoA carboxylase. That means formation of malonyl-CoA is dictated by citrate. Correct? This is the thing that you remember. Citrate allosterically stimulates acetyl coenzyme A. What is next? Now, high level of citrate will be actually active when we have a lot of ATP. That means we should be in well-fed condition. We should be in well-fed condition. What is well-fed condition? When we are eating enough in stipulated time gap. Enough. Like we are eating 4 times or 5 times, 4 times, but we are eating only half of the roti. But we are eating 4 times, we are eating 1 packet of biryani 4 times a day. Which one is well-fed condition? The second one, correct? So when we are eating more than what we require, ATP is abundant. And that triggers the citrate to stimulate acetyl coenzyme A, acetyl CoA carboxylase. This enzyme is very, very important. This is allosteric regulation. What is next? এগুলো ছেড়ে দাও, দরকার নেই। এত ডিটেলস দেওয়ার দরকার নেই, জাস্ট রিমেম্বার অনলি ওয়ান স্টেপ। আচ্ছা, what is the end product? Permitted. Is there any role of permitted here? Where is permitted? Permitted. See the action of permitted. Now, kindly explain, anyone explain, what do you mean by allosteric and covalent modification? How does it differ? Is there any difference? Here it is showing covalent modification under allosteric regulation. What is the difference, I want to ask you? Anyone? এই ও ডাক, তোর টাকা আছে? টাকা নেই? আছে কি নেই? এখন নেই? তোর কাছে টাকা আছে? হ্যাঁ, বের কর, টাকা নিয়ে আয়। তোর কাছে টাকা আছে? আয়, ও এত সময় নিচ্ছে, গরিব মানুষ। হ্যাঁ, দেখ, সরকার নিয়ম করেছে যে টাকায় যদি আমি পেন দিয়ে লিখে দিই, সেই টাকার কোনো থাকে না, তাই না? পকেটে রাখ। এবার দেখো, allosteric regulation. সিগারেট আছে ওই যা কিনে নিয়ে আয়, এই টাকা দিয়ে কিনতে হবে। আচ্ছা, মনে কর দোকান আছে, তুই মনে কর ওই দোকানদারকে দে, ও মনে কর দোকানদার, ওকে যদি দাস এই টাকাটা দিস, ওকে সিগারেট দেবে? যা দেখ নেয় কিনা দেয়। আচ্ছা, আমি মনে কর দোকানদার, আমাকে টাকাটা দিই, এই কে সই করেছে? এই কাজ চলবে না, অন্য টাকা দাও। আমার কাছে সিগারেট আছে, ওর কাছে টাকা আছে, আমি অ্যালোস্টেরিক মডুলেটর, ও এনজাইম, কারেক্ট? আমি যেহেতু কোভ্যালেন্ট মডিফিকেশন করে দিয়েছি, তার ফলে আমি ওকে ধাক্কা মারে পাঠালেও এই টাকা দিয়ে ও রিয়াকশন করতে পারবে না। ঠিক আছে, টাকাটা দিয়ে টাকা কিছু লিখিনি, পরিষ্কারই আছে, টাকা নষ্ট করিনি। যা, কথা বুঝতে পারলি কি বললাম? একটা এনজাইম যখন ফাংশন করে, কাজ করে, তখন তার দুটো জিনিস লাগে, এক ইচ্ছা, আর দুই শক্তি বা সামর্থ্য। কোভ্যালেন্ট মডিফিকেশন ফসফোরেশন বা ডিফসফোরেশন করে এনজাইমটাকে উইক বা স্ট্রং করে, আর অ্যালোস্টেরিক রেগুলেশন সেই এনজাইমকে সেই ইচ্ছাটা দেয়। অ্যালোস্টেরিক রেগুলেশন মানে কি? কান টানলে মাথা আসে। আমি তো মাথায় হাত দিচ্ছি না, আমি তো কান ধরছি, দেখবি কান টানলে মাথা আসে কিরকম? অ্যালোস্টেরিক রেগুলেশন আজ উঠায়। হ্যাঁ, দেখ একটা অদ্ভুত ব্যাপার, আমি ওর কানটা ধরে মুছে দেবো, কান চিৎকার করবে না, মুখ চিৎকার করবে আর চোখ দিয়ে জল বেরোবে, দেখবি? দেখবি? দেখ দেখছিস? যা, তাহলে এইটা হচ্ছে অ্যালোস্টেরিক রেগুলেশন। আমি টানলাম কান আর কাঁদলো চোখ, বোঝা গেল? হ্যাঁ, তাহলে কিন্তু আমি যদি চোখে লিকোপ্লাস্টার লাগিয়ে দিই, মাইক্রোপোর লাগিয়ে দিই, তাহলে চোখ কাঁদবে? কাঁদবে না। তাহলে ওটা হলো কোভার্ট মডিফিকেশন, বোঝা গেল? তো যখন আমি বলছি এরকম একটা রেগুলেশন হচ্ছে, তখন দুটো জিনিস খেয়াল রাখতে হবে। এন্ড ফিডব্যাক রেগুলেশন হয়, ফিডব্যাক রেগুলেশন হয় সবসময় এন্ড প্রোডাক্টে। হোয়াট ইজ দ্য এন্ড প্রোডাক্ট? পারমিটেড। So permitted will react with where? Acetyl-CoA carboxylase is active when it is dephosphorylated. কি করে মনে রাখবো? খুব বেসিক, একদম সেই গ্লাইকোজেন মেটাবলিজমের শুরুতে বলেছিলাম। There are two enzymes, glucagon and insulin. They always work against each other, no matter what, just like India and Pakistan. No, sorry, Israel and Palestine. ঠিক বললাম? না, তাও না। আচ্ছা, ইউএসএ অ্যান্ড রাশিয়া? হ্যাঁ, এরকম টাইপের সবসময় বুঝি যে গ্লুকাগন করে কি? ফসফোরাইট করে। ইনসুলিন করে কি? ডিফসফোরাইট করে। তো যে সমস্ত রিয়াকশন অ্যানাবলিক রিয়াকশন, সেগুলি অ্যাক্টিভ হয় এনজাইমসগুলো ডিফসফোরাইট হলে। অ্যাক্টিভ অ্যাসিটাইল-CoA কার্বক্সিলেজ ইজ ডিফসফোরাইটেড ফর্ম অফ দ্য অ্যাসিটাইল-CoA কার্বক্সিলেজ। তার মানে এফেক্ট অফ ইনসুলিন, কারেক্ট? পারমিটেড কি করে? দেখ, পারমিটেড কি করে? এই অ্যাসিটাইল-CoA একে ইনহিবিট করে। মানে পারমিটেড কিছু অনেক তৈরি হয়ে গেছে, জমে গেছে, সে তখন ফিরে গিয়ে একে বলে ফিডব্যাক মেকানিজম। সে কি করবে কি? একদম শুরুতে ফিডব্যাক মেকানিজমের দুটো শর্ত আছে। এক, এন্ড প্রোডাক্ট। দুই, রেগুলেটরি পয়েন্টটা হতে হবে at the early stage of the metabolic pathway. দেখ, লাস্ট প্রোডাক্ট পারমিটিক অ্যাসিড, আর কোথায় কাজ করছে? অ্যাসিটাইল-CoA কার্বক্সিলেজ, দ্য ফার্স্ট রিয়াকশন। দুটোই ফুলফিল করছে। সো ইট ইজ আ ফিডব্যাক মেকানিজম। It is a feedback mechanism. Now, how does it take place? By two methods, allosteric regulation and covariant modification. What is covariant modification? Removal of phosphate group or addition of phosphate group. What is allosteric regulation? Working on the acetyl coenzyme A not being a substrate. Is palmitic acid a substrate for acetyl coenzyme A? Carboxylase? No. But still, it will act on acetyl coenzyme carboxylase. That's why it is called allosteric. Clear? So glucagon, epinephrine will and insulin will stimulate. Hello? Hormonal. Next. It's called amphotericin. What is it? Hydrophobia, hydrophobia. Yes. Hello. এই পাতা hydrophobia, এই পাতা hydrophobia. এবার original synthesis পাঁচটা স্টেজে পাবো। স্টার্টিং এ যে কম্পিটিটিভ অ্যাক্টিভ হয়ে যায় কি করে পাবো? সেম ম্যাথমেটিক্স সিস্টেম থেকে বাইরে আসবে, সিস্টেম থেকে অ্যাক্টিভ করতে হবে। The cholesterol biochemistry is expressing these 5 stages. Number 1, 5 stages. Number 1, formation of mevalonate. Formation of mevalonate. Number 2, formation of isopenoid side chains, isopenoid units. Number 3, formation of squalene. এটা কি হলো? 5 স্টেজ আমি লিখেছি না, এটা পরে কোথা থেকে আসলো ওটা? Isopropyl units formation of squamine. Formation of lactosterol. For non-cholesterol, what we have to remember from here is structure of cholesterol, then stages of cholesterol biosynthesis, then only the first stage. That's all. Very fast. This is a reaction. Please say it. Please say it. Start writing the steps up to development and we say 3 years. This is probably the only pathway that contains maximum number of molecules. Almost every state is discovered by one of the molecules. Number 2, these pathways produces enormous number of intermediates which participate in various types of reactions. Right? So, এই হলো first reaction that we do is cholesterol synthesis in all cells. Cholesterol is very, very necessary for our body. Cholesterol is never bad. Cholesterol is good. We make cholesterol bad by our sedentary lifestyle and bad habits. Number 1, smoking. Number 2, drinking. These 2 bad habits plus sedentary lifestyle makes cholesterol bad. Otherwise, cholesterol is a food, a nutrient for our brain. For our brain. You have good amount of cholesterol, your brain is active. And normally it prevents premature death. What is going on? Some papers have been published to show cholesterol prevents premature death. So, accident enzyme is then from accident enzyme forms ATP weight, beta hydroxypropanone chloride weight. Enzyme is ATP weight synthase. Both the state, one of the enzyme is removed. One of the enzyme is removed. So 3 molecules of the same enzyme contains to form 1 molecule of ATP weight or beta hydroxypropanone chloride weight. The right way. आगे बार हम निकला right way 3 carbon dihydroxylic acid इतना बोला 5 carbon dihydroxylic acid the right way 5 carbon dihydroxylic acid the right way right so this is right way is HND way next stage is different into different pathway for photosynthesis into different pathway
retroduodenal and dividing into 2 branches, right gastroepiploid and superior pancreaticoduodenal. Right gastroepiploid lies along the greater curvature of the stomach.
Disconnected.
And superior pancreaticoduodenal, it is supplying head of the pancreas and second part of the duodenum. Okay, so these are the
Connected.
different branches and their divisions and supply of the 3 branches of the celiac trunk. Okay. Next. So these are the branches.
Disconnected.
Gastric artery giving esophageal branches and gastric branches. You remember the previous picture, okay? Common hepatic artery, you practice those previous pictures. That is one schematic diagram, okay? So common hepatic artery, it is giving gastroduodenal artery dividing into right gastroepiploic, superior pancreaticoduodenal, right gastric artery and hepatic artery proper. Hepatic artery proper again dividing into right and left hepatic artery, right hepatic giving cystic artery. Okay, and this from the gastroduodenal also giving the retroduodenal and supraduodenal arteries for first part of the duodenum. Then splenic artery, it is giving pancreatic branches, arteria pancreatica magna, arteria pancreatica cauda, short gastric arteries, left gastroepiploic artery, which is giving gastric and omental branches. So these are the branches of the celiac trunk. Okay. Next. Okay, now we will discuss the individual branches. Left gastric artery, it is the smallest branch of celiac trunk. It runs upward behind the peritoneum towards the cardiac end of stomach. Here it arches forward and enters the lesser omentum, and further it runs along the lesser curvature of the stomach. It gives 2 to 3 esophageal branches at the cardiac end of stomach to supply the lower part of the esophagus, and gastric branches along the lesser curvature, and it anastomoses with the right gastric artery in the lesser omentum. Next, this is left gastric giving esophageal branches and gastric branches. কে কথা বলছিস পিছনে? উঠে দাঁড়া তো, এতবার ধরে বারণ করছি শুনতে পাস না? বলছি তো ক্লাস না করতে চাইলে বেরিয়ে যা, অ্যাটেন্ডেন্স দিয়ে দেওয়া হবে। Okay, previous. Next is common hepatic artery. It passes downward and to the right behind the lesser sac under the cover of parietal peritoneum, and it reaches the upper border of first part of duodenum to enter into the lesser omentum. And here it gives gastroduodenal artery and then continues upward along the free border of lesser omentum as the hepatic artery proper. Okay. Next, gastroduodenal artery. It arises from the common hepatic artery at the upper border of first part of the duodenum, runs downward behind the first part of duodenum and divides into right gastroepiploic and superior pancreaticoduodenal. Then right gastroepiploic artery, it arises from gastroduodenal artery at the lower border of first part of the duodenum, and it runs to the left in the greater omentum along the greater curvature of the stomach. It gives omental branches to the greater omentum and gastric branches for the stomach, and anastomosis with the left gastroepiploic artery in the greater omentum. Okay. Next. See, so this is gastroduodenal dividing into superior pancreaticoduodenal and right gastroepiploic. Okay. Also giving 2 branches, supraduodenal, retroduodenal, and passing behind the first part of duodenum. You can see this is dotted line. Okay, dotted line means lies posteriorly. Next is superior pancreaticoduodenal, arises from the gastroduodenal artery behind the first part of the duodenum, and soon it divides into 2 branches, anterior and posterior. And these branches run downward in corresponding pancreaticoduodenal groove that lies between the head of the pancreas and second part of duodenum, and ends by anastomosing with the branches of inferior pancreaticoduodenal artery. Okay. Next is right gastric artery, arises from common hepatic artery at the upper border of first part of the duodenum, enters and runs to the left along the lesser curvature of the stomach, ends by anastomosing with the left gastric artery. Okay. Next is hepatic artery proper, which is continuation of common hepatic artery after the beginning of gastroduodenum. It runs upward within the free margin of the lesser omentum to the left of the bile duct and anterior to the portal vein. And in the porta hepatis, it divides into right and left hepatic arteries to supply corresponding physiological lobes of liver. Okay. Next. See, this is the hepatic artery proper dividing into right and left hepatic. Right hepatic is giving cystic artery. This is right gastric artery and this is gastroduodenal. These are the three branches from the common hepatic. Okay, understood? Next is cystic artery. It arises from the right branch of hepatic artery at porta hepatis and it passes downward and to the right behind the common hepatic artery and cystic duct to reach the neck of the gallbladder. And here it divides into superior inferior branches to supply the corresponding surfaces of the gallbladder. Okay. Next is supraduodenal artery of Wilkie. It is an occasional branch of gastroduodenal, supplies upper margin and adjoining surfaces of proximal part of first part of the duodenum. Okay. Next. So you can see this is supraduodenal artery of Wilkie, commonly asked in your Viva and MCQ. Okay, branch of gastroduodenum. And this is cystic artery dividing into two branches. Okay. Next, splenic artery. Splenic artery is the largest. This is very frequently asked. Tortuous artery. What are the other tortuous artery in our body? This is very frequently asked. Facial artery, uterine artery. Okay, these two are also tortuous. Splenic artery is the largest branch of celiac trunk. It is tortuous, passes to the left behind the peritoneum along the upper border of the pancreas. Okay, this is location along the upper border of the pancreas. It crosses anteriorly to the left suprarenal gland and left kidney, enters the linorenal ligament, ends by dividing into 5 to 7 splenic branches. Okay. On its way, it gives pancreatic branches, left gastroepiploic artery, short gastric artery, and splenic branches. Next is pancreatic branches of splenic artery. These are arteria pancreatica magna. This is largest pancreatic branch of splenic artery, accompanies the main pancreatic duct. Then arteria chordae pancreatica, it is a large pancreatic branch of splenic artery that supplies the tail of the pancreas. And dorsal pancreatic artery is branch of splenic artery, runs downward on the posterior surface of pancreas and divides near the inferior border of the pancreas. The left terminal branch is also called transverse pancreatic artery. Next, so see, this is tortuous splenic artery, dividing into 5 to 7 splenic branches, giving short gastric artery for fundus of the stomach, giving left gastroepiploic artery. These are the pancreatic branches. Largest pancreatic branch, arteria pancreatica magna. Okay, and this left gastroepiploic, it is anastomosing with the right gastroepiploic along the greater curvature. Okay, next is short gastric arteries, 5 to 7 in number, arise from terminal part of splenic artery, and these branches supply the fundus of the stomach. Left gastroepiploic arises from terminal part of splenic artery, runs in the gastrosplenic ligament. Splenic artery runs in the linorenal ligament, and this left gastroepiploic, it runs in the gastrosplenic ligament. Okay, and greater omentum along the greater curvature of the stomach. It gives gastric and omental branches to supply the stomach and greater omentum, ends by anastomosing with the right gastroepiploic artery. Celiac trunk and their branches commonly show variation in their beginning and their course. Okay, next. So this is left gastroepiploic, right gastroepiploic. Next. Okay, that's all. Okay, কোনো query আছে? Celiac trunk is very important. It can come as your short note. Okay.